Hello everyone. Today we will discuss about tRNA or transfer RNA. It is also known as soluble or sRNA. These tRNAs are relatively small, single-stranded and globular molecules, having molecular weight 2 to 3 into 10 raised to 4. These tRNA molecules are made by the action of RNA polymerase 3. Then coming to the structure of tRNA, secondary structure of tRNA is known as clover leaf structure or clover leaf model. tRNA molecules vary in length from 75 to 90 nucleotides. In eukaryotic cells, 10 to 20% of nucleotides of tRNA may be modified, and these are known as unusual nucleotides. Unusual nucleotides. For example, dihydrouracil, pseudouridine, ribothymidine, etc. These are the unusual nucleotides in tRNA. So coming to the structure of tRNA, the secondary structure is known as clover leaf model or clover leaf structure. All tRNA molecules contain 4 main arms or loops. 4 main arms or loops. So, the tRNA molecules, it contains 4 main arms or loops. That is D arm. This is D arm. Here it is anticodon arm or A arm. This is T arm. Here acceptor arm or A arm. So, first we will see D arm. So, D arm, it contains the base dihydrouridine. That's why the name D arm, dihydrouridine. It has 4 to 6 base pairs. 4 to 6 base pairs. Then coming to A arm, that is anticodon arm. This is unpaired and non-bonded loop. This is anticodon. Anticodon. This is unpaired and non-bonded loop, and it carries specific sequences of 3 bases, and it constitutes anticodon. 3 bases and it constitutes anticodon. The bases of the anticodon in tRNA are hydrogen bonded with 3 complementary bases of codons of mRNA. These are hydrogen bonded with complementary bases of codons of mRNA. So, this anticodon arm contains 5 to 7 base pairs. 5 to 7 base pairs. The next arm is T arm or T loop. This is also known as T psi Z. T psi C. Psi is pseudouridine. So, thymine, pseudouridine and cytosine. Thymine, pseudouridine and cytosine. This is T loop or T arm. It contains 5 base pairs. 5 base pairs. The next one is acceptor arm or A arm. Acceptor arm. So this consists of unpaired sequences of cytosine, cytosine, adenine. Cytosine, cytosine and adenine at the 3' end. The 3' end is the acceptor end. So the 3' hydroxyl terminal of adenine, 3' hydroxyl terminal of adenine, it may bind with the carboxyl, alpha carboxyl group of the incoming amino acid or specific amino acid. So this is the amino acid binding site. So the 3' OH terminal of adenine bind with alpha carboxylic group of specific amino acid and it forms amino acyl tRNA complex. Amino acyl tRNA complex. This is for protein synthesis. And this acceptor arm contains 7 base pairs. 7 base pairs. So these are the 4 arms or loops of tRNA. There is an extra arm that is known as variable arm. It is most variable structure of tRNA and it forms the basis of its classification. Mainly class 1 and class 2 tRNA. Class 1 and class 2 tRNA. Class 1 and 2. About 45% of tRNA belongs to class 1 tRNA. And they have 3 to 5 base pairs in its extra arm. For example, alanine tRNA. About 45%. Then about 25% of total tRNA belongs to class 2. It has 13 to 21 base pairs in long chain. For example, phenylalanine tRNA. So based on the attachment of amino acids in the 3' end, tRNA is classified into 20 types. Based on the attachment of amino acids in the 3' end. This tRNA is classified into 20 types. Then tRNA constitute 10 to 20% of total RNA. This is a secondary structure that is known as Clover leaf model. Then a tertiary structure of tRNA, it is L-shaped. It is L-shaped. This L-shaped tertiary structure is formed by further folding of the clover leaf due to hydrogen bonds between this D and T arms. The main function of the tRNA is it helps to decode a mRNA sequence into a protein. It helps to decode mRNA sequence into a protein. tRNAs function at specific sites in the ribosomes during translation. Then this tRNA is linked to a particular amino acid and base pair with a codon in mRNA so that the amino acid can be added to a growing peptide chain. This tRNA is linked to a particular amino acid and base pair with codon in mRNA so that the amino acid can be added to a growing peptide chain. Each tRNA molecule is recognized by one of the 20 amino acyl tRNA synthetase enzyme. So, this is about tRNA, that is transfer RNA. It is also known as soluble or sRNA. So, this is about tRNA. This is today's topic. Thank you for watching.